Hello and welcome to this video on membrane transport. In this video, we'll be discussing different types of membrane transports along with their examples in much detail. So let's dive into the lecture. Membrane transport is a fundamental process in a biological system which allows for the movement of molecule across the biological membrane. We know that cell membrane is a selectively permeable membrane that separates extracellular and intracellular compartments. To maintain the homeostasis, there must be some mode of exchange between these compartments, without which cells cannot survive. This exchange is made possible by the two processes. One of them is the passive transport, another is the active transport. Let's discuss them in much detail. What is passive transport? Starting with it first. Passive transport is the movement of molecule from their higher concentration to their lower concentration without input of any energy. Okay, so molecules are moving downhill, so there will be no use of, or there will be no input of energy in this case. Because molecules are moving downhill, the process or the molecular movement will be spontaneous and random in this case. Molecules will move randomly in this case. So this passive transport includes simple diffusion, facilitated diffusion, and osmosis. These were the examples of passive transport. Now let's move to the another process which is known as active transport. Active transport is the movement of molecule from their lower concentration to their higher concentration. So the process is against the concentration gradient. There must be some expenditure of energy in this case, without which process cannot occur, without which movement cannot occur. So this active transport includes primary active transport, secondary active transport, receptor mediated transports. So these are the examples of active transport. We will discuss them in the preceding sessions. Right now we are going to start with passive transport, and one of the passive transport is the simple diffusion. Let's start with the simple diffusion first. What is simple diffusion? It is the random, you must remember, it is the random movement of molecule from their higher concentration to their lower concentration, from their higher concentration to their lower concentration without input of energy. So because there is no use of energy in this case, there is no input of energy in this case, so the process will be spontaneous. Let's try to learn through this diagram. The oxygen is taken up by the cell by the process simple diffusion, because you can look into this diagram that in the extracellular fluid the concentration of oxygen is higher, in the intracellular fluid the concentration of oxygen is lower. We know that simple diffusion is the movement of molecule from their higher concentration to their lower concentration. What all substances can move by the process simple diffusion? Substances which are smaller in size. Substances which are smaller in size, having low molecular weight, and above all, substances which are lipid soluble can easily cross the membrane by the help of process known as simple diffusion. So just revising it again, what all substances can move by the process simple diffusion? Substances which are smaller in size, having low molecular weight, and above all having lipid solubility. These conditions are required in order for substance to move through the process simple diffusion. Okay, so there are some factors which affect the rate of simple diffusion. Let's discuss them one by one. Simple diffusion is directly proportional to the concentration gradient. Greater the concentration gradient across the membrane, greater will be the rate of diffusion. So rate of diffusion is directly proportional to the concentration gradient. Next is the surface area. Suppose this is the membrane A, which has got smaller surface area, and this is the cell membrane which is B. Okay, so having larger surface area, the rate of diffusion or the movement of molecule in case of B will be at higher rate than the A. It's because of the surface area. So the rate of diffusion is directly proportional to the surface area. Third factor is the solubility. Solubility, greater the molecule is lipid soluble, greater will be the rate of diffusion. So the rate of diffusion is directly proportional to the solubility. The fourth factor is the thickness of membrane. If membrane is more thick, the rate of diffusion will be less in this case, because molecule have to travel longer distance in this case. So rate of diffusion is inversely proportional to the thickness of membrane. The fifth factor is the molecular mass. Greater the molecular mass of molecule, lesser will be the rate of diffusion. So rate of diffusion is inversely proportional to the molecular mass. Okay, what all substances can move through the, what are the examples of those substances which can cross the biological membrane by the simple diffusion? These substances are some gases such as oxygen, carbon dioxide, nitrogen, some fatty acids, and alcohol, which is highly soluble in lipid, so it can easily cross or it can easily move by the process simple diffusion. But there is the problem with the simple diffusion. Those molecules which are larger in size and which are having charge, they cannot move or they cannot cross the biological membrane by the process simple diffusion. So there is the another process known as facilitated diffusion, which help them cross the biological barrier. Large molecule includes glucose, some amino acids, okay, and charged ions includes sodium ion, potassium ion, calcium ions. Okay, so you can clearly see in this diagram, these are the glucose molecules. Glucose molecules are transported by the help of glucose transporters, also known as glut transporters. These transporters are of 14 types. Of these 14 types, glucose 4 transporter, GLUT4, is activated by the insulin. Okay, the GLUT4 is activated by the insulin. When insulin bind with its receptors, these receptors are tyrosine kinase in this case. Okay, so these are the tyrosine kinase receptors. As soon as insulin bind with their receptors, there will be the autophosphorylation of tyrosine molecules in these receptors. And after some signaling pathways, we know that within the cell there are some vesicles which contain GLUT4 transporters. After insulin stimulates these receptors, these GLUT4 transporters now gets translocated within the cell membrane. Okay, now these transporters allows the movement of glucose from the extracellular fluid to the intracellular fluid. These GLUT4 transporters are mainly present within the hepatocytes, within myocardial cells, within muscle cells. Okay, so this was about the movement of glucose through the GLUT transporters. These GLUT transporters also makes the movement of galactose and mannose possible across the membrane. Okay, so they also help the movement of galactose and mannose. There are some channels which are made up of protein, contain tunnels. Okay, so these are the channels which contain tunnels which are water filled. Through these tunnels, ions can move across the membrane. These channels could be sodium channels, they could be calcium channels, they could be potassium channels as well. There are some channels which contain gates. These gates are sensitive to the voltage. We know that normally in the most cells of the body, the resting membrane potential is minus 90 millivolt. When this potential reaches to the minus 60, these gates are opened. Okay, so these gates are now opened. By the opening of these gates, ions can move across the membrane. Okay, so these channels are known as voltage gated channels. They could be voltage gated sodium channels, they could be voltage gated potassium channels, they could be voltage gated calcium channels. Okay, there are some channels which are not sensitive to the voltage, but they are sensitive to the ligand. When their ligands such as neurotransmitters, such as hormone bind with their domain, these channels are opened, which allows the movement of ions across the membrane. And there are some channels which are not regulated by anything. They are opened all the time. So they allow the movement of ions across the membrane all the times. They are the leaky potassium channels. We studied in the simple diffusion that with elevation of concentration of diffusing substances, the rate of diffusion was increasing proportionately. But this is not true with the facilitated diffusion. With the increase in the concentration, the rate of diffusion, facilitated diffusion, also increases till the point where maximum rate is achieved. What happens at maximum rate? Whatever receptors, whatever channels were present, whatever transporter proteins were present within the membrane are now saturated with the diffusing substances. So there won't be further increase in the rate in this case. Okay, so this must be clear to you. Because of the saturation, there won't be increase in the rate. No matter you increase the concentration, the rate will remain same, which is the V max in this case. Okay, so let's write it, which is the V max in this case. That is the maximum rate.